Okay, I'm going to start from the beginning. So good morning, everyone. It's a pleasure to see all of you. And this is the ideal way to meet during this period, in particular because our guest is arriving from Shanghai. So it's a real pleasure to have invited Professor Hui Yang to join us for the APPD Family Center for the Prevention and Treatment of Parkinson's Disease monthly lecture series. So we usually meet on campus, and in fact, we had invited Professor Yang to join us in January, and he was about to arrive, and then he had to postpone because he received a prize from the Chinese Academy of Sciences, a Young Scientific Prize. And so he postponed to March 12th, and well, the rest is history, and you know now why he wasn't able to join us on March 12th. So we're extremely fortunate that we can host Professor Yang. And let me tell you a little bit about him, give you some background. So Professor Yang did his first degree in Shanghai at Glia-to-Neuron University. He then did his PhD at the Shanghai Institute for Molecular Biology at the Chinese Academy of Sciences, and then went on to do a postdoctoral fellowship for 2 years with Professor Rudy Jaenisch at the MIT. He returned then to China not too long ago and has really done remarkably well. In addition to gathering prizes up through January, his expertise is to study human diseases by creating CRISPR mouse models, and he's also moved into non-human primates, and his goal is to get to gene therapy. He's had a really remarkable 3 years. He's had papers, and I hope that many of you have gotten a chance to look through them, in Nature, Nature Methods, Science, Nature Neuroscience, Genome Biology. And the topic of today is going to be a mouse model for Parkinson's disease and other neurodegenerative disorders. And this was a paper that just came out a few weeks ago in Cell. So on behalf of Professor Nir Giladi and I, co-directors of the APPD Center, it's a pleasure to host you, Professor Yang. And please, if you would share your screen, and we'd like to hear your talk.
Yes, thank you for Karen's introduction and invitation. I'm so sorry I couldn't come in January, so I couldn't go face-to-face talk. But luckily we could go using Zoom in this special time. So I will show my screen now. Wait a second. So good morning, everyone. It's my great pleasure to show our work about how to using gene editing approach to treat Parkinson disease and other neurodegeneration disease while transdifferentiation from non-neuron cell type to specific neurons. Since this is first time I give a talk for a center, especially for special disease. So because I assume you are familiar with Parkinson's disease than me.
We seem to be stuck, so please, if I can just have everyone's patience while we sort this out.
Sorry.
Welcome back.
Okay, sorry, just a call come back. Sorry about the interruption. And for neuron degeneration disease, for such as Parkinson disease, it will specifically the internet is not worked well.
Yeah, the practice worked very well.
Yeah.
Okay, a few more minutes of patience, everyone, and we'll
Sorry.
We'll get it working.
Yeah. It worked. Okay, let's come back. Scientists also usually use two approaches for stem cell or treating this disease, such as stem cell therapy or in vivo directly transdifferentiation. For stem cell therapy, people also usually transplant this dopaminergic progenitor cell into the patients. This cell could divide from fetus or human embryonic stem cell or iPS cell. However, the most challenge of this approach is this progenitor cell will get a very low survival rate, or how this cell get integrated into the local neural circuit. Another is the common concern for this stem cell therapy approach is immuno rejection and tumor genesis. So alternatively, people like to get in vivo transfer. People like to get in vivo direct reprogramming non-neuron cells type such as astrocytes into the neuron. We could just simply or overexpression X1 or neuroD1 or neuroD2 to get the astrocytes into the neuron with very high efficiency, usually over 90%. However, we couldn't get the dopaminergic neuron type. Recently, a group from Sweden, they using 4 factors, 3 translation factor and 1 microRNA. Through overexpression 4 factors, they successfully get the dopaminergic neuron, but with very low efficiency, usually about 10 to 20 TH positive cells in 1 slice. And you should deliver multiple factors via AAV virus. So you should packaging 2 or 3 AAV virus. You should ensure one astrocyte infected with these 4 factors simultaneously. So it's pretty hard. So because our lab is focused on gene editing approach, so we asked whether we could using gene editing to get this transdifferentiation. For DNA editing, you could using Cas9 or other family to get the DNA gene knockout or gene correction. And for RNA editing technology, you could using Cas13 to just simply break down regulation of target mRNA. But for the epigenetic editing, you could mutate with the nucleus domain, and you will get a data Cas9. They could easily bind the DNA double strand, but they don't get the nucleus activity. So you could fuse with activator domain such as VP16, or you could fuse with inhibitor effector such as KRAP. You get the gene expression inhibition. So using this system, we could get the simply gene overexpression or inhibition. So because usually we should overexpression one or multiple transcription factor from one cell type to another cell type. So we choose this system for our further research. We first try to improve this system, get a very high robust efficiency, activation efficiency in vivo. So we optimize this approach by combining several effectors, such as we combine the Cas9 with some tag system. Using this system, we could get multiple effector using one guide RNA. Also, we besides VP16, we also fused with P65 or HSF1. So using this SPH system, you just need one guide RNA. You could get multiple activator in the gene promoter of the gene. So you could get a very robust gene activation in vitro. Then we put this system into the mice because they are very large size. So we get the transgenic mice. So if we want to activation one gene, we just simply get one guide RNA, packing into the AAV virus, and we could get the gene activation. It works very well for single gene activation. If we package this guide RNA into the multiple array, we could get multiple gene activation simultaneously. So next, we test whether it works well could it be used for in vivo transdifferentiation. Then as previous study, we using GFAP mRNA to label specific labeling astrocytes, and using guide RNA to activation genes such as AX1 or neurodegenerative simultaneously. After injection AAV virus, we isolated the injection region and found all three genes could be activated simultaneously. And we also successfully get the astrocytes into the specific into the mature neuron. You could see if without activation, the GFAP mRNA virus will specifically label it with astrocytes. But after activation AX1 and nucleotide 1, you could see the neuron-like cell emerge, colocalized with mRNA signal. So in this work we prove using CRISPR activation system, we could in vivo simultaneous activation of multiple genes, and also by this gene activation we could directly change from astrocytes into neurons. However, the challenge for this approach for clinical application is the delivery of this system via AAV virus, because this system have very large size. We need 2 or 3 AAV virus to deliver into the human patients if we apply this approach for in vivo for clinical application. So we
Professor Yang, if I can interrupt a minute. we're not seeing the whole slide. So it might be better if you're not in presentation mode. We'll see the background of your PowerPoint, but at least then we'll see the whole slide because it's cut off for everyone.
Okay, great. That's fine.
Thank you.
I will. Yeah.
Yeah, this way we see the whole slide.
Okay, great.
Thank you.
So you could, yeah, okay. I will next. So we next asked whether is there any gene knockdown or knockout could also get in vivo chance differentiation, because using gene editing system, it is more easy to get a gene knockdown or multiple gene knockdown. Even multiple gene knockdown, it's easy to packaging gene editing tools into one AAV virus. But for gene activation, you should fuse with activator, so there is larger size. It's very difficult for clinical application. So we search the literature for is there any gene expressed for just for knockdown or knockout could get in vivo chance differentiation. Luckily, we found a work from Dr. Xiangdong Fu's lab. They published this work in 2013. They found just a knockout one gene, PDB1. This is a microRNA binding protein. This work shows just the knockdown of them with shRNA. They will get multiple non-neuron cell type into the neuron. They proved this work very efficient, demonstrate this work very efficiently in culture cells, including such as HeLa cell, N2A cell, or MAC cells. However, we don't know whether it works in vivo or also whether we could induce the specific neuron type in vivo. So we next want to test it. Whether before do this, we compare with shRNA approach with our CRISPR-RNA testing approach. We could find using gene CRISPR-13 systems such as CasRx is Cas13D. They have smaller size, less than 1 kb, less than 1,000 amino acids. So it would be easily to pack it into one AAV virus, and it works very robust compared with shRNA or CRISPR inhibition system. And also they compare the specificity of this system. They found it's very highly specific compared with shRNA approach. So we decided using this approach for our further experiments. At the same time, we also proved this system works very well in other systems such as the liver or retina. When we inject this AAV virus into the mice, we found that we could efficiently knock down such as PCSK19 or VEGFR target gene efficiently. And for VEGF-A, we treat this. We proved it in age-related macular degeneration. So next, we want to next we want to ask whether it could works for PDB1 knockdown. To do this, we first design 6 guide RNA, 2 guide RNA for each paragraph. Because just for 2 guide RNA, we could easily pack the name into one AAV virus, and we assume more guide RNA will work better than 1 guide RNA. We could get efficient knockdown efficiency. And then after screening in N2A cell and all astrocytes, primarily astrocytes, we could get CasRx with guide RNA 5 and 6 with better efficiency than knockdown. And also after whole transcriptional sequencing, we found it's highly specific, both in N2A cell or astrocytes. So next step, we want to decide which region we should inject it. For Parkinson disease, people usually inject a virus or transplant the stem cell into the SNC or striatum. Because we think for SNC, even they could get induced dopamine neuron, they should projection the axon into the striatum and release the dopamine. So, alternatively, some scientists also inject into the striatum for stem cell therapy or in vitro differentiation. They assume that the induced dopamine will just locally release the dopamine. They don't need to form the axon. So, for our proof of concept study, so we decide to inject our AAV virus into the striatum. Then for we first using as previous we use the GFAPM cherry for specific labeling exercise, and for CasRx we also using GFAP promoter driven CasRx. It could be ensure this gene editing tools just expressed in the exercise, not in other type of cells. It could ensure the safety of this approach. Then after injection, 1 week later, we get the slides and get the staining. We found the astrocytes could be specific labeling with this GFAPM cherry virus, and also the CasRx was specifically expressed in the astrocytes. And also 1 week later, we get the slides and the staining found the PDV1 was specifically locked down in the astrocytes and with a very high efficiency. Then 1 month later, we analyzed the mice with and found about over 50% of the astrocytes will turn into the neuron. And we further analyzed the subtype of these neurons, and we found about half of them is excitatory neuron, not the inhibitory neuron, because they express glutamate. Also, some very low percentage of this induced neuron will express the interneuron marker, but we fail to observe the PV neurons. And interestingly, we also observed the TH positive neurons. It indicated that maybe we could use this approach to treat the Parkinson's disease. Because you will see this picture, there will many fibers from SNC neurons, and so it is very hard to distinguish which one is your induced neuron, which one is fibers. But because this is we analyze them with the confocal, you could analyze each slice and see you could easily you could clearly distinguish which one is your induced neuron, which one is the fiber. Although by this approach we could found a small percentage of the induced TH neurons, and but less than 10%. So we want to test whether in Parkinson's disease model this efficiency will be, whether it will be increased or not. So we next try to prove it in the Parkinson's disease model using OHDA infusion. 3 weeks later, we could see the TH positive neuron will be lost in the SNC region, also in the striatum. And so after 3 weeks, as 3 weeks infusion, we inject the GFAPM cherry and our CasRx system into the striatum, and about 1 month later, we could see clear many positive TH neurons, also dopamine neurons with DART positive neurons. This is a major dopamine neural marker, and we could get you could compare with the Y type neuron. You could see the efficiency will be increased, and also after 3 months, the percentage will be increased with the time. So. However, we couldn't see the TH or DART positive neurons in 1 or 2 weeks. So we think from exercise to the mature neurons, TH positive neurons, it need about 3 or 4 weeks, at least 3 or 4 weeks. We then calculate the absolute number of TH positive cells, and we found the number is increased, and we could find about 200 TH positive cells per section. Compared with previous work, I mentioned before, for each publication in Nature Biotech, they're using 4 factors. They only could get just around 10 positive TH cells. So our efficiency is increased more than 10-fold just using single factors. Next, we analyze whether this TH positive neurons is like SNC neuron or like VTA dopamine neurons. So after staining, we found these neurons much more like with SNC neurons, but not VTA neurons. They don't express the VTA dopamine neuron marker. And also, whole cell recording found most of them will have electrophysiology, and 4 out of 10 neurons will show delayed voltage rectification. It is a marker of TH neurons. And also, we test whether this dopamine neuron could release dopamine. We found this effect we met. This channel for dopamine release, and also it could release and uptake dopamine. So this induced the neurons with. We proved this induced the neurons with dopaminergic features in Parkinson's disease model. Finally, we analyze the, so I will, because this video, I will show your screening. Finally, we analyze the behavior. We just analyze them for 1 month. I started with the 1 month. We could observe the obvious significant improve the disease motor dysfunctions, which directly induced, including apomorphine and also amphetamine induced rotation. You could say just 1 month, you could say obvious and significantly alleviate the motor dysfunctions in the treated group. Also, we also analyze them in 3 months. They also get very improved function behavior. We also next finally we analyzed the two drug free motor dysfunction behavior, and we including cylinder test and the rotator test. Also, we analyzed them just 1 month, and we could have found obvious obvious improvement of this treated mice. After our just at the same time, Dr. Xiangdong Fu's lab also put his work into the preprint journal bioethics. They different from us. They inject the AAV virus, also knock down PDB1. They using shRNA approach. They inject virus into the SNC. They found it could also efficiently induce the dopaminergic neuron, and this neuron will send the exon send the projection and into the striatum and release dopamine. But the behavior rescue, at 1 month they couldn't get the recovery of this, but at the 3 months they could see obvious disease phenotype rescued. So we because in 2 regions for striatum and the SNC just knock down PDB1, we could get efficiently transdifferentiation. So we asked whether this could be worked in other systems, not in the central brain. So we next test them in the retinal system. We using this model with RGC loss, rich ganglion cell loss, we treated it with the drug, and then after several weeks, we inject the AAV virus to target the mular cells. We found that it could efficiently get regenerated rich ganglion cell. And surprisingly, this rich ganglion cell will send their projection into the central brain, and not just form the axons. They will send the projection in the correct place, such as in contralateral. They will send more axons, and ipsilateral they will get few axons. And this step is one step by step. They will send first. We will observe the name in contralateral, and then we will see in ipsilateral. And this regenerated ganglion cell will alleviate the symptoms of this disease, and using VP or just functional behavior test. Simultaneously, in other group also post their work in preprint. They found by PDB1 knockdown, the mular cell could also regenerate into the cone cell or also in the road cells. So by PDB1 knockdown, the cell type is depend on the disease environment, and also your injector regions. So finally, I want to discuss what we should do next. First, because our lab is gene editing, focus on gene editing approach, we think whether we could optimize our gene editing tools. As you see, this AAV virus is about 4.3 kb. Close to the limitation of AAV virus. So if we could get a smaller gene editing tools, we could be easily to packaging them and easily to go to clinical application. Another is we only test our systems in about 2 or 3 months old mice. However, for neurodegeneration disease, it usually occurs in older patients. So it would be very interesting whether it also works efficiently in the older mice, such as 1 year old or even older. Another is whether it works in other systems, because our disease model works only for drug-induced. It have no disease environment. So whether it works in genetic, the Parkinson disease mice model, and also whether it works in stroke or asthma disease, and also spinal injury. It would be very interesting to be tested. And another is our lab's interesting is whether it works in non-human primate. We using drug or surgery, we could easily get a non-human monkey model with glaucoma or Parkinson disease. And currently, we are now focusing on it. If it works, I think this approach will very close to clinical application. So finally, I want to thank our director, Dr. Muminpu, give our much support and advice for this project. And I will want to thank three members in our lab, and two postdocs and one PhD students. He already got a job for our investigator. And also thanks our collaborator in our institute, because we are expertise in gene editing, but we have very few experience on the neural technology, such as EEG or whole cell recording in vivo. Yeah, they give us a very good support. And thanks for the funding. And last, thank you for your attention. Yeah.
Professor Yang, thank you so much for that really exciting presentation. It's remarkable how much you've done in really such a short amount of time. So we can open up for questions. And so if anyone, I can see Professor Korchin and Professor Horowitz. So Amos, please, you have a question? And you can unmute yourself.
Okay, thank you very much. You hear me now?
Yes, yeah, very well, thank you.
Thank you very much. This is really very, very impressive. And I'm admiring the scientific achievement and also what you've done so far in terms of Parkinson's disease. However, let me just say that Parkinson's disease is, of course, a neurodegenerative disease. And it's not just the nigral cells which are affected. So how do you go about it? You know, it's not just the dopaminergic cells. In fact, the dopamine system is not that important in terms of therapy, because we don't need your cells to replace the dead cells that will provide dopamine. We have levodopa, and levodopa does it very well for a while, and at least. But the question is, and of course, the nerve cells, the neurons that you will provide us with, the patient with, of course, do not have the connections that regulate them. So it's not just having the nigral cell replenished, but it's how do you make them work in a normal way?
Yeah, I think yeah, this is just the first step. We treat the patients with levodopa, but the dopamine neurons, I think, is not just release levodopa. They maybe will get other, they will maybe release the other important component. We don't know. So I think the next step, I prefer if we could get the Negro, they will get the correct projection. I think maybe it's the better way, but we don't know whether it could form the exon correctly in older patients. We just say for Shandong Fu's work, they just into the nebula, inject AV into the nebula, and they will get the projection into the striatum. But our work is just inject into the striatum. Yeah. You should. It would be interesting to compare with these two systems. But for the mice model, I don't think it's the good model because it's hardly to analyze the cognition, higher behavior, just the motor behavior. Maybe the large animal model, we will get more information. Yeah.
Professor Horowitz, please, you have a question.
Hi, beautiful, unbelievable system. Good luck. And you look so young, so you have many years, and I hope that you are going to really do tremendously well. So my question is talking about Parkinson's disease, non-idiopathic, non-genetic Parkinson's disease. Something is wrong in these dopaminergic cells, and for some reason they die. There's misfolding, there's, God knows, something is wrong in the cells in this patient. And you are now replenishing the, let's say, a specific area of the brain with the dopaminergic cells. But once they die, because there's something wrong there, and so these new cells may not live as long as you wish them to live, but they will die from the reason that the other cells, the former cells, the older cells, died as well.
Yeah, but I think this is a good question. But you could say, I think the die, you need time. Yeah, maybe, yeah, you need time to die. Maybe you could get a 1 year or 2 years die. We don't know. But for these works, they prove after this is genetic disease. For the eye disease, they get the RPE lost. But after regeneration, this regenerated RPEs could survive maybe for over 1 month or 3 months. Yeah, they have the disease niche. Maybe they still need a time to die. Yeah, I think in the future we maybe need to combine with some drugs, and you also get the correct cells. Yeah.
Thanks.
Yeah, thank you.
Hi, thank you for this beautiful talk, and I would like to follow up both Mia and Amos' question. Practically, what you have shown us that you are developing a new symptomatic therapy for advanced Parkinson. And do you think your technology can be taken into a more early stage preventive approach?
Prevention?
Yeah, using that in a much earlier stage of Parkinson, not at the time that they are unable to move, and you're bringing some new approach to improve locomotion or movements, but using that technology for early stage Parkinson, and for either intervene in the basic mechanism of the disease.
Yeah, I think it's our very good question. Is our major interest, but I think for clinical trial, maybe we could only practice this approach in a later stage of patients, and if it is safe, we step by step for early stage patients. Yeah, because after you get the motor dysfunction, about 30 or 80% of the dopamine neurons are already lost. Yeah.
Thank you. Moore Cohen has a question. Moore, I'm going to unmute you. Okay, there you go, Moore.
I thank you for this talk. It was amazing. I want to ask about the off-target effect. Did you check it with this treatment? And also, looking at the future, and you're talking about better methods of editing for human treatment, I think it must be discussed, the off-target effect as part of safety.
We only analyze them in which culture system. I think it's pretty hard to analyze in the vivo system because it's a trans differentiation. Cell type will get changed, and the translation profile will get changed. So we get changed. So it's not like gene editing. Once cutting, they will retain. But for RNA editing, I think we will still analyze. We try to improve the safety. But for in vivo analyze, I think it's pretty hard for current stage. What I think for off-target, I think you could see, I think it's better than shRNA approach. Obviously, because in culture system we already proved. But in vivo, I think, yeah, it's the big issue. We should establish some experiments to demonstrate it is safe. But compared with the DNA editing approach, I think RNA editing approach is more safe. Another we want to claim is once this exercise go to the neuron, the gene editing tools will be turned on because we using GFAP promoter. It's only expressed in the exercise. Once they go to into the neuron, the GFAP promoter doesn't work. There will no oxygen editing. So once editing, so it's just expressed a short period of time, maybe 1 month. Yeah.
Thank you. Professor Uri has a question.
Hi, thank you very much for the great talk. I have a question to one of the slides you showed at the end regarding glaucoma. You mentioned that you can now, produce retinal ganglionic cells. So, and maybe I missed something. Are they functional? Have you tested it in, the more functional ways or just the markers?
This way, we got the function way. This glaucoma.
Yeah.
Okay. We get the projection and we get the EEG recording in the brain, and also do behavior test.
Okay.
Dark light behavior test. Yeah.
And this was after 1 month of differentiation.
Yeah. Well, after 1 month, after 1 month differentiation is enough.
So you have growth of new neurons into the to the visual thalamus and visual.
Yeah. Just 1 month, I see it's crazy. Yeah, it's surprising. Yeah, I was surprised. It's formed very quickly. Yeah, and we calculate the absolute number of this regeneration, such as Y type, you will get 100 RGC cells, but for the regenerated tissue, you will get about 20 cells. Yeah, 20 cells I think is enough for the functional recovery. Yeah.
Thank you. Does anybody else have any questions? I don't see all of you, so that unless you send me a chat, I won't be able to see a question. Any more questions? Okay, so if you do have a further question, you can email me and I'll forward your email to Professor Yang, and then you can discuss it directly with him. So thank you so much. I really hope that you will have the opportunity to visit us in Israel. One of the other things that you're going to do is give us a workshop on CRISPR editing, because it's something that, of course, we're very keen to improve our skills on. So again, I want to thank you very much for joining us all the way from Shanghai. I want to thank all of you who joined us this morning, and we'll be in touch and let you know about our next monthly series.
Yeah, thank you. Thank you for coming.
Thank you. Bye, everybody.
Yeah. Bye. Keep in touch. Okay. Bye.
השאלה של קודם מאיה, שאתה